So our first presenter today is Chuck Call. Dr. Call is the Chief Technical Officer for Bioflyte. Dr. Call has 30 years of startup experience working in bioaerosol sampling, detection, and microbial identification. He earned his PhD in mechanical engineering at UC Davis and has over 40 US patents issued. So I want you to welcome Dr. Call.
Thank you, Wayne and Greg and all those who helped organize the event. I'm happy to be here to give an update on Bioflyte. This is our technology, but I want to start with just a little bit of review of what we got done last year. In April of last year, we closed on a Series A financing, about $6 million. So that gave us some runway to get moving, and we did. We brought in May. We recruited and brought in a new CEO, Todd Sickles. He's from Santa Fe. Some of you may know Todd. He was supposed to be here today, but he's out in Washington, D.C. working with some potential customers today. And so he asked me to step in for him, which I'm happy to do. But Todd has hit the ground running in May and has really done a spectacular job with getting the company aligned and pointed towards the customers. And it's going really well. In the fall, we standardized our mass spec product, which you see here. And so we're now moving into production mode, and I'll get to a little bit of our plans in 23 here in just a minute. So just real quickly, what we're doing is time of flight mass spec. It's a MALDI instrument. If folks are familiar with MALDI, time of flight mass spec, Bioflyte is really the first to bring this technology from the lab to the field. And this is a fully integrated robotic system that samples the air, analyzes the sample, and resets itself. There are essentially no consumables. There's a stack of cartridges in there, but we can run up to 200 tests between refresh cycles. So it does a full microbial identification of a bioaerosol sample. Does all that in about 5 minutes. Oh, I see. For the online folks, sorry about that. A little detail there I wouldn't have known about. So hopefully everybody can see fine. So you can see kind of the instrument has the BioTOF module is on the far left, and on the far right or in the middle is the aerosol sampling, and on the far right of this platform here is sort of the cartridges for the sample disc. So the instrument packaged up looks like the gray box there, and is meant to operate in the field. Right now, we have some units in mailrooms in New York City doing biothreat screening for things like anthrax, and I'll talk a little bit more about that. And we have a unit that's a couple of units that are going into a couple of airports in Q1 of this year for counterterrorism applications, and in particular bioaerosol and biothreat detection. It's fast, under 5 minutes, and species level specific. So that's something that's never existed in the marketplace. It's something that can be fast, essentially affordable to sustain out in the field, and not needing a lot of consumables, PCR reagents, things of that nature, but providing a species level answer. So moving on, let's see, there we go. There we go. Okay, so as I mentioned, the markets that we're going after really is attentionally occurring microbes and threats in the air. In particular, we've been focused on biothreats, things like anthrax and ricin, which you occasionally see in the news. And also we've been, recently we've demonstrated that we can sort of a proof of concept at this stage, not integrated into the product just yet, but we can detect airborne fentanyl and other opioids essentially in near real time, and again, without the use of any consumables. So essentially we get that for free if we're doing the biological analysis as well. So that's additional capability we're trying to bring to the product. There's also naturally occurring microbes in the air, as we all know, having just been through the pandemic. So there's potential, especially in the healthcare settings, to move this type of technology into monitoring for pathogens in the air in hospitals and manufacturing facilities of pharmaceuticals and things of that nature. So those are really the markets and target customers that we're doing. The mail screening product looks a bit like the upper picture. It's we've integrated our mass spec with a workstation where a mailroom clerk can operate a mass spec, believe it or not. So there's really just nothing to do but push the button, and everything else takes care of itself. It's fully automated and autonomous and connected to the cloud. And so we see all the data in real time and provide reach back. So that's sort of the markets where we've had this installed since last July at one of the big banks in New York City, and that's converting to revenue right now. And we have about 5 more target customers that we're planning to hit over the first half of this year. And in the lower half, what we're showing is really bringing the technology to critical infrastructure protection. And in particular, we're focused on transportation infrastructure and airports in particular. And we have, as I mentioned, a couple of these units going into airports. One of them will be here in Albuquerque. The other one isn't public yet. And so but we have a big target market and a good opportunity, we believe. A lot of the funding for the development of the technology, not MALDI time-of-flight mass spec, obviously, but this miniaturized version that's integrated with the aerosol components. A lot of that funding has come from the DHS, Department of Homeland Security. There's an office there called CWMD office, Counter Weapons of Mass Destruction. They funded a lot of it, and then DOD through the DTRA, or the Defense Threat Reduction Office. So those are have provided a lot of support over the years. Talk a little bit about 23 and sort of our roadmap. First up, you can see is a facility move there on the upper left. We've been with the Bioscience Center since it opened, I guess. They're one of Stu's inaugural clients, and I'm happy to say, and Stu's probably happy to acknowledge, that we're graduating. We're not one of the fastest, but it's a complex product, and it's taken a while to get to this point. But we've got 4 units in production in there right now, and we're kind of bursting at the seams, and some of the younger companies are looking to grow and would like our space. So we found a new space over in the Sandia Science and Technology Park, and we'll be moving there hopefully in the next week or 2, God willing, right, Bob? Yeah. They're struggling with the tenant improvements, but we're hoping that all gets worked out here like in the next 24 hours, and we're able to move in a week or so. So yeah, we're pushing them as hard as we can. So and so that's a big deal. We're looking to expand our workforce probably by a factor of 2 this year. We have 4 positions open, from entry level manufacturing technician positions, really building mass specs, on up through director, VP level manufacturing operations, and some manufacturing engineers in the middle of that. So definitely looking to do some hiring. Go to our website if you know anybody who's looking for work. We're certainly trying to fill 4 positions right now. I'm in charge of the manufacturing tech position. And so shoot me a resume, or jump on the website, even better, if you know somebody who would be interested in that position. So things are looking good. We're closing on some financing right now. I don't have any details on that, but Todd will shortly, I think, and that's going very well. And we actually, it's a tough environment for raising money, so we're very excited. And I think, as I said, Todd hit the ground running, and we're going to have a lot of great things happening this year. And so Todd will bring those out at the appropriate time. Thank you very much.
Very good. Thank you very much, Chuck. Appreciate that. Just a couple of questions I have on this. So your units that you have, how large a room will a unit be required?
So the units actually get placed not in the room, they get placed in the mechanical room where the return air goes. So we basically typically would put one unit in one HVAC or one ventilation zone in a big building. So the Albuquerque Airport, for example, has 19 zones and would require 19 systems. There are some ways to cut back on that. And there's some trade-offs in terms of how fast and how much of the building you could protect. One of the things we've shown is that if you know it's there and you modify the action of the ventilation system, you can mitigate the spread. We've also shown that if you don't do that, it spreads to the entire building over about 2 hours. So there's a lot of benefit. And we're also, one of the things we want to do late this year is start coupling in exposure modeling. So if we know, and really the benefit of the system in terms of ROI, in terms of exposures to such an event.
This is going to change a lot of infrastructure to have to, because if they have a centralized location where it's reading all the rooms, right? And let's say, and I don't know how this machine determines what room Hello? Okay, there we go. Thank you. So you have that central location. This unit is installed. So you have 20 rooms in the building. This is going to tell you that building A is where the germ is located, correct? It's going to tell you what room it is. And then once you locate that room, what do we have in place? Because you're going to have to quarantine that room. Who's going to come in and take care of that? Because I don't think we have the infrastructure in this country right now. This is why this could be a foundational sort of thing. Who's going to come in and take care of that virus that's invaded that one room? It can't be the police, it can't be the SWAT team, right? You have to have BioTech people come in. So I'm just kind of curious, like, how is that rectified? You know, just
I'll answer the question two ways. In the context of an airport, it's not by room, it's by ventilation zone. And if you get an anthrax attack in an airport, the airport shut down and you're going to spend $10 to $100 million cleaning that up, maybe hundreds of millions if you don't contain it at all. Because it'll get to other airports and everything else. So there's a lot of benefit to knowing it happened and containing it, but it's a huge job, and at least a portion of the airport will be out of commission for a while. In the context of a room, a classroom, this technology is light years away from trying to be able to be affordable at the point where you could put it in and do room by room surveillance. So we're not there yet. That's another generation away. And so, but we have decon technologies, spray fogging systems that have been developed by others. And for example, Build with Robots, a company here in town, has demonstrated a fogging machine out at the airport that could come in and disinfect at night, things like that. So that technology is out there, but our detection technology isn't ready to be deployed at that kind of a level.
I don't really know what New Mexico has in place, but again, if this is located at the airport, who do we send in at New Mexico to take care of this? Or is this on a federal level, or is this a state level that we have something in place, the experts that take care and clean up, I should say?
There's protocols for all that, regardless of how an event like that is detected. There's protocols, emergency management, probably manage the incident. And yes, the federal folks will get called in. As
this
needed.
is invaluable technology. At least we know it's there. You stay away from that area, right?
Yeah, exactly.
Very good.
Great,
Thank you.
thank you. Any other questions for Chuck? Yes, sir.
Yeah, maybe a follow-on to that question. Is there a way to identify the source of a pathogen? So for example, if you have maybe multiple devices, could you triangulate that, hey, you know, that person over there maybe has a briefcase full of fentanyl or something? Could you find out where it's coming from?
Most likely the way that would be managed is through cameras and looking at the video and observing the behavior associated with it. You're not going to be able to run around quickly enough. I mean, we'll detect it within 5 minutes or something, and then you can quickly look and see, well, who is likely candidates. You can probably narrow it down to a couple people through suspicious behavior and other things.
And maybe a second question, are there any other devices that do something similar, or what kind of device are you guys trying to supplant? Or is this completely unique in the market? Nothing else is available?
It's fairly unique. The Pentagon, for example, has a system. There is a technology out there that's consumable based, and they're expensive, like on the order of $100 an assay. And they typically combine this with maybe some particle counters and other things. and run their assay when they suspect something is going on. So because the consumable is expensive, they set the thresholds pretty high. Because our cost per test is essentially zero, you can set the thresholds a bit lower and have better protection. So there is a product out there. It's not widely deployed because of the cost. And so it's really, you wouldn't think cost would be the reason why there's no protection in any airport in this country. But it is because there's only so much money and resource to go around. And I think cost becomes an important factor.
Sounds like a great unmet need. Thanks, Chuck.
Thank you. Any other questions? We have time for maybe one more quick question.
Thank you. Is this generator driven? In other words, if the electrical grid goes down, will this be running while the lights are out, let's say, or is there a backup system to keep the thing functioning if the electrical system in the building shuts down, let's say? Because it happens.
Yeah, got it.
Periodically.
This machine, believe it or not, only uses about 60 watts of power,
Yeah.
less than a light bulb. So we can run a UPS uninterruptible power
Okay.
supply. So battery backup, you
Yeah.
can run for hours.
So this is a 24/7, 365 watch, basically.
Exactly.
Yeah, very good. Okay.
Very good. Thank you very much, Chuck. Appreciate that. And just so glad to hear that you are looking to hire people. That is very good news for New Mexico. So our next company, thank you. Our next company is T-Neuro Pharma. Dr. Kristina Trujillo is the CEO of T-Neuro Pharma. She was a professor and researcher at the University of New Mexico School of Medicine for over 10 years. She's published numerous peer-reviewed articles, received grants from NIH, American Cancer Society, and the DoD. She has completed a sabbatical at Great Point Ventures, which is a VC firm in San Francisco, in which they go out and try to identify emerging technologies that look like worthy of further development. She's now an entrepreneur, and she looks for these emerging technologies and tries to bring them into the real world. So, Kristina, you may proceed.
Thank you for the introduction, Wayne, and thank you for inviting me. We've had a lot of movement in the company lately, so I'm really excited to share our progress. So T-Neuro Pharma is an Alzheimer's company that's taking a completely different approach to Alzheimer's because we're using T cells to track and treat the disease. So Alzheimer's has both a diagnostic and a therapeutic problem. Diagnostics are costly, they're invasive, and most of the time only detect the disease after damage has been done to the neurons. Therapeutics have been 99% ineffective, and the ones that do sneak by the FDA are incredibly expensive and arguably ineffective as well. The root cause of both these problems is the same, in that they've targeted amyloid beta and tau, which makes sense because these are the proteins that make the plaques and tangles in the Alzheimer's brain. But those are just symptoms of an earlier causative event. And T-Neuro has data that supports we've identified one of those earlier causative events. Dysfunctional T cells go into the brain, attack the neurons, and cause Alzheimer's pathology. We have human clinical data showing that we can detect the level of these T cells in a blood sample, and that can identify patients early in the course of the disease. That's the basis of our T-Track diagnostic. We have animal data showing that we can block these cells from going into the brain, and that prevents Alzheimer's pathology. And that's the basis of our T-Treat therapeutic. Now, some of the feedback that we've gotten when we show this slide saying we've identified a causative event is that's a mighty big claim for a little bitty company. What makes you think that you've identified this when these billion-dollar companies haven't been able to? So I'll go through a little bit of our data that supports this giant claim. Oftentimes, there's three criteria that are used to show that an agent causes a disease state. The first one is adding the agent to subjects induces the disease and the disease pathology. So we've published data showing that we can introduce these abnormal cells into a mouse. The cells go into the brain, they attack the neurons, and they cause all of these human-like Alzheimer's pathologies in this model. So this is important because this is the very first time anybody's been able to show a causative event that induces these pathologies. And whenever you look at the two most targeted proteins, amyloid beta and tau, you can see that even those proteins that they've been spending billions of dollars on aren't able to induce all of these pathologies, whereas our T cells are. So the second criteria is that you have to be able to inhibit the agent and prevent the disease in the model that you caused it in. So we go back to our model, and we've published data showing that we can block these cells from entering into the brain. And we can also inhibit the function of the cells to where they can get into the brain, but they can't attack the neurons. And that actually inhibits all of these Alzheimer's-like pathologies from happening. So these two criteria can only be investigated in animal models because obviously you can't induce something in a human. So to see how it translates to humans, we have to show that the agent is present, or at least preferentially present, in diseased patients. So we looked in the brains of Alzheimer's patients, and we find a statistically significant elevation of these cells in the brains of Alzheimer's patients. We've repeated this in multiple cohorts with multiple different markers of the T cells, just to be sure. And the result is repeatable. It's statistically significant each and every time. So we've met all three criteria of causation, which to us is a very big breakthrough, but it's not very useful as a diagnostic because you can't go searching around in live patients' brains for these cells. So we wanted to see what these cells were doing in the circulatory system. So we did flow cytometry to identify the cells in the blood, and what we found was that the level of these cells actually correlates very strongly with the progression of the disease. And as a biomarker, this can reach 85 to 90% accuracy. Now, to put that into perspective, the gold standard for diagnostics would be cerebrospinal fluid biomarkers. But keep in mind that involves a spinal tap. It's painful, it's invasive. Not a lot of people are going to want to do it, especially for a yearly screen. And we can reach the same level of accuracy with a simple blood sample. So our long-term vision is to use the T-Track diagnostic to screen the general population over 55 yearly. This will identify the patients most likely to develop, to develop the disease, at which point we can administer the T-Treat therapeutic, preventing them from ever getting the disease to begin with. So we've divided the plan into multiple stages. The first stage is to develop the T-Track diagnostic, and the goal of that stage is to have a standalone diagnostic with actual clinical revenue. The second stage is to do all the preclinical studies that are necessary for the IND application and approval from the FDA. Now, we believe will be an attractive acquisition target at the end of either of these stages for some of these larger biotech companies and pharma companies who really are desperate to bring new innovation into their pipelines. So right now we're focused on stage 1, developing the T-Track diagnostic. So the revenue model is to be reimbursed by Medicare, Medicaid, and insurance. And based on similar in vitro diagnostics, we anticipate roughly a $300 reimbursement. The market is the 40 million Americans who get annual blood screening, and with a $300 reimbursement, that works out to be a $12 billion total addressable market. But until this gets recommended as a yearly screening test, our market is the 500,000 Americans diagnosed annually. And because we're starting in New Mexico, the market is the 5,000 New Mexicans diagnosed annually. So right now, patients have available to them PET scans or CSF biomarkers. Now, those are expensive, they're invasive. There's also other blood biomarkers that are under development, but these are also expensive. And the most important thing to remember is that ours is the only one that's targeting a causative event. So the scientific milestones we've reached, we've published 2 peer-reviewed papers, and we have a 3rd in preparation for submission to Science. We've shown the T cells cause Alzheimer's pathology. We've shown their potential as a diagnostic, and we've shown their potential as a therapeutic. Our business milestones include securing an exclusive license from Cedars-Sinai for both diagnostic and therapeutic use. We've been accepted to the New Mexico LEAP program, which secures $400,000 in non-dilutive funding to develop the T-Track. And we've also obtained a $1 million commitment for our Series A round once we identify a lead investor for this round. To date, before our company formed, our chief scientist, Dr. Wheeler, raised $1.2 million in grant funding. We currently have a convertible note open with a $4 million cap, and we have $200,000 remaining on this note. Once we close that note, we'll open our Series A, which is a $4 million raise specifically to develop T-Track. The $200,000 on our open note is going to be used to secure international patent protection and to complete a study comparing amyloid PET to our biomarker. The Series A is going to be used to develop our diagnostic as a laboratory developed test. Now, LDTs are important because they're usually the first step a diagnostic will go through before it gets into widespread use, and it does not require pre-market approval from the FDA. So we anticipate having this as an LDT in New Mexico clinics within 2 to 3 years, at which point we'll start looking for acquisition. So you heard about my background. The rest of the team is Dr. Chris Wheeler. He comes from Cedars-Sinai. He has over 30 years of neuroimmunology and clinical trial experience. John Chavez has successfully exited multiple tech companies. And Wayne, as you all know, he comes from Pharma and he has a lot of experience in Alzheimer's programs. Thank you. I'll take any questions.
Thank you, Kristina. Are there any questions from the audience?
Oh, prevent the cells. So we have 2 strategies. One is to attack a molecule called CD103, and that's a molecule that allows it to cross the blood-brain barrier. And that's our strategy that we've done so far. The other strategy is to actually inhibit the ability of the T cells to secrete interferon gamma, which makes them completely ineffective. So those are the 2 targeting strategies.
Have you done research to know if this is genetic or environmental when these T cells begin to invade? You know, is that genetic? Is it environmental? What do you think?
So it's likely not genetic because Alzheimer's, the genetic mutations are only associated with about 5% of patients. And those are usually the early onset patients. So we don't know for sure. I can't answer that 100%, but most likely it's environmental.
Do you know the environmental factors that might feed into it? Because, you know, we've got lack of exercise, fruits and vegetables, you know, this is what we're hearing all the time. Make sure you take care of yourself.
I mean, it's a lot of speculation. There's a lot of different studies to put together, but there is some suggestion that infection has something to do with it. I don't want to go into that story a lot. My chief scientist would probably wring my neck for speculating. But there is a little bit of data pointing in that direction.
So we know broccoli is not causing it.
Probably not.
Okay, good. I love broccoli.
Any other questions for? Go ahead.
I just wanted to understand, you don't need pre-market FDA approval, but when will you need FDA approval? At what point does that come in?
So we'll need FDA approval if we want it to be a yearly recommended test. So we'll get it into the market. If there's enough data that doctors feel like they can prescribe or that they can order it and they'll get useful information, they can do that. But along the way, we are going to be generating the data so that it is recommended as a yearly test.
Hi, Kristina. That was a great presentation. So my question is more on the science side, as far as what classifies a T cell as quote unquote dysfunctional? Is it the accumulation of damage? I know you were looking at aged T cells. So what does that look like?
Yeah, so these specific T cells, what we classify as dysfunctional is the surface markers. I mean, that's how we identify them. But these specific ones will attack the amyloid precursor protein in the brain. And so that specific class of T cells is what we consider because they're attacking that protein.
Okay. Any other questions? If not, thank you very much, Kristina. Our next presenter is Dr. Sergey Dryga, and he's the founder and CEO of BioTech. Dr. Dryga is experienced in developing products for in vitro diagnostic and vaccine markets, custom services, and government contracts, skilled in leading all aspects of R&D and product development that requires combination of molecular biology, protein engineering, and analysis, organic chemistry, immunology, virology, bacteriology, and other ologies, and the interface with electrical software and mechanical engineering. And he's building a company that will build scientific instruments. Dr. Dryga?
The company is really new. I received my articles of incorporation in November last year. But it's based on about 30 years of experience in the lab. One problem that we see always in the lab, practically any biotech research involves some form of purification. Protein purification is very common. For nucleic acid purification, methods became more available and easy. But for protein purification, we are really stuck with two primary methods. One is you buy a reagent set, like in high school, and you do purification manually. You spend about 2 or 3 hours tied to centrifuge or drip column. And if your protein is heat sensitive, you do this in cold room. It's a lot of fun to spend a day in cold room. Other approach is you buy a chromatography system that is fantastic instruments. They can do a lot of things. They result in very pure protein, but they cost, they begin entry-level system at about $70,000. Decent system will cost about 150. And you need to spend about 3 months training to use that system well. And then after you have done all of that, it will still cost you a lot of money to purify. And since it's a very expensive system, when you change proteins, you have to clean it every time. So at one point, when I was talking with my scientists in my lab, he said, let's do a system that is automated and single-use cassette. So if your lab looks like pictures on the left, these are actual running purification systems from 2 different companies. Then welcome to the future of purification. On the right is prototype automated purification system. So the instrument, I called it Inceptum, fully automated platform for purification. You put your sample on one side in a vial and take a purified protein or RNA or DNA from the other vial, and your hands-on time is about 5 minutes. Put sample in and take it out. In the meantime, you can take coffee break or smoke break or whatever break, and or do another experiment. The system uses, it's fully functional chromatography system. It uses 2 columns. It has 2 sets of detectors for optical density and for conductivity, which are 2 primary parameters that are measured in chromatography systems. So if idea is that good, it must have market size. We look at some market projections or estimates. Basically, chromatography purifications, pretty decent size, about 6 billion. Our total addressable market is about $1.4 billion. And this is US only. My strategy right now is to enter US market and worry about other markets later. Primary customers are researchers in research institutes, academia, and biotech and pharma companies. So at some point I went to a business school and they told me to always put McKinley chart. So yes. On x-axis is purification time. The further to the right, the faster it goes. And on y-axis is cost of instrument. Right? And the higher up, the less expensive it is. And circle diameter is cost per purification. And most of the systems have comparable costs for purification. But what sets BioTech apart is speed. And speed and also other benefits like we have cost comparable to manual system, but purity of the protein, quality of the purified material comparable to big chromatography systems, because we are a chromatography system. So our value proposition is purified protein right now when you need it. A business model is basically printer and ink. The cost of instrument, I kind of have to set some price for it, but I can give it for free and still be able to make money on the cassettes. My initial kind of cost of the instrument would be just below CapEx limit for most organizations, so that it goes as a consumable. You don't have to get no departmental approvals to buy that piece. Manufacturing of the instrument will be in-house, assembly from parts, from outsourced parts. It's a relatively simple system, but it also, development of that system resulted in 2 pending patent applications and 1 in process. So team, right now it's me. I have a couple of partners who provide engineering support services. In terms of my background, I have some experience in biotechnology. I've done about 15 years in vaccine development and another 15 in vitro diagnostic systems. Have some 30-ish patents in technologies, in semiconductor and electronics. And 2 partners, Digital Health Solutions and other product solutions, help me with engineering parts and software cloud integration of the system. Because this is a device that will be hooked up to cloud, so you can monitor when you are on your coffee break, you will know when it's done. So I would really like to have beta testers. If you run lab, talk to me, and let's get system in your hands, and you'll play with it and tell me what's wrong with it, because there should be something wrong with it, and it will help me develop a better system. And of course, being startup, I'm looking for Series A investment. once prosecute patents, have 2 pending applications and 1 in submission, and start manufacturing product. If you scan this code, it will get you my contact information, or you can stop by a table and
Very good, very good. Thank you very much. Yes, sir.
I wanted to ask you, with this device, what would you say is the #1 market in the hospital itself? Is that what you're targeting? Hospital?
research, not hospital.
I'm sorry?
It's a research institutes.
Research Institute.
Yep.
Okay, so tell me real quick, how would I benefit as the public?
As a public, let's say you're developing COVID test, right? If it takes 3 days to run through protein purification, and you save 2 days out of this 3, that's faster time to market.
It's going to tell me that I have COVID.
No, this will help develop test.
I see.
It doesn't do tests by itself, and therefore I don't need FDA to give me approval to sell it. The only thing I need to do is to make sure that my device doesn't electrocute anybody, and that's it. And I can sell without too many approvals.
Thank you.
Any other questions for Sergey? Sergey and I were chatting a little bit before the presentation. So no regulatory approvals are required. It's simply getting it into the hands of users, getting it published and out into the market. Is that right?
Yeah, that's correct. Yep.
Very good. Thank you very much. Okay, our next presenter is Dr. Janeth Pena. Dr. Pena is the flora body, I want to say antibody every time, flora body specialist and chief scientific officer at Sandia BioTech. She is skilled in genetic and protein engineering. Janeth also has a broad background in invertebrate biology and genetics from her thesis work, where she studied the interplay between schistosomiasis and their snail host. Sounds really interesting. So I want you to welcome Dr. Pena.
Thank you for the introduction. Well, I want to start by saying that I work for an antibody producing company. Funny side note, snails don't have any antibodies. But it's been a wonderful experience. And so just a little bit on Sandia BioTech, we specialize in producing reagents that can accelerate the research of scientists. However, for this particular talk, I'm going to focus only on our fluorescent reagents. As you can see in the images here, I'm going to start with our split GFP and super folder GFPs. Now, this is technology that we license from Los Alamos National Labs. And it's a very powerful technology because when it comes to the super folder GFP, these proteins were very difficult to produce previously. And with some of the modifications that the scientists did at Los Alamos, now you have a protein that can fold incredibly fast, but can also help other proteins fold faster or more effectively if there's issues as scientists are trying to work with proteins. Now, why do we want to work with proteins? Well, they do everything. Well, almost everything, right? And so we want to see how they're interacting with one another. We want to see maybe what is being switched on or switched off. All of this can be monitored through protein-protein interactions. And so one of the developments that came from the super folder GFP is actually the split GFP. So the idea that you can take one component of that protein, and then once it comes in contact with the other 10 components, as we see here, 1 through 10, then you have a fluorescent molecule. And this is very beneficial for several reasons. You can have proteins that are linked to either side or to either ends of these proteins. They help them fold, and then we can track them and we can tag them. The reason I mention this is because after COVID and everything started getting open, we actually got a lot of interest in this particular product of ours. So we've been working closely with another company that's doing high throughput development using our product. And that's become very beneficial for us. And we're seeing that this is something that's going to continue for 2023. When it comes to being a reagent company, high volume is really what you need in order to have a successful company. We have other fluorescent technology, and this is the one that I'm going to focus on for the remainder of the talk, which is the fluorobody technology. And this we license from UNM and from Queen Mary University. Now, what's really interesting, or what I particularly love about this particular product, and it's because I am a molecular biologist by trade, is that we can create these structures so that they can be beneficial for a variety of different applications. So at this time, we actually have 100 plus fluorobodies developed, and these are antibody analogs, and they can be used for, to benefit the scientist in life cell imaging and flow cytometry. And I'll go into detail in a little bit on what a lot of those are. So one of the benefits of these molecules, they're smaller, so they can reach places that other molecules can't. We've changed their structure to where it cuts protocol times. So it's saving time for the researcher, and they are inherently fluorescent. So there's no need for additional labeling of the molecules that then affects how well they can bind. It's quantitative by nature because you have that unique 1-to-1 ratio, the binding site that will find your protein of interest, and then the molecule that is sending the signal for you to track it. There's no cross-linking, so that just means that as you're doing the experiment, you are not, like making other things happen that then you would record within the cell. And then we have it in multiple colors, so there can be multiplexing. We make them in bacteria, so we may have to talk for the purification and maybe the beta testing. And so I just want to share with you some of the things that we've done when it comes to the fluorobody technology. When COVID happened, and this is a little bit of a continuation from the talk that the president of Sandia BioTech gave last year, Tony Pena, we focused on creating partnerships with other startup companies, and so that we can showcase the strengths that we have. And so one of those companies was Etaluma, from which it's a immunofluorescent microscopy company. And it's a wonderful microscope because it's very small, it's very simple to use. And so you can set it up in a variety of institutions and students can use them. And one of the tag phrases that we ended up coming up with is labeling and imaging made simple, because our product makes protocols very simple and their microscope makes just the viewing of the cells very simple. And so here we're actually together at the NM Pavilion from the Bio 2022 conference. And we actually got a lot of traction because everyone likes looking at pretty colors and cells fluorescing. I do want to add, and I forgot to mention this in my previous slide, as you see the green fluorescence of the cells here, that's actually from the super folder. So we were able to merge the two technologies that we have licensed, right? One from Los Alamos, which is a super folder protein, and the technology that we have from UNM, and now we have a super folder fluorobody. Other collaborations that we focused on when it comes to just getting our name out there, because we have tested our product, it is validated. And so we just need to get our advocates so that people know that we're here. We partner with Protein Fluidics, and they have an amazing device that allows the grow of tumoroids in a very simple and fast way. But one of the issues that their clients have is actually the ability to stain the tumoroid effectively. And because our product or our molecules are smaller, our proteins, then they can penetrate better into the tumoroids and label efficiently. Oh, I think I may have, okay. So keeping it local, we are also working with a flow cytometry company here called NMBio. This is a startup, and they're looking at entering the market for high volume testing for flow cytometry. So in flow cytometry, you can use it for diagnostics, and our product lets you know which cell is which. So if you have a cell that is Jane and you have a cell that it's Bob, our product lets you know who is Jane, who is Bob. If you have less of Jane, you may have HIV. If you have more of Bob, maybe you have cancer. Sorry. And so that's one of the things that we're focusing on in testing here. We are also working on other projects related to flow cytometry when it comes to a local reference lab here. And when it comes to the different applications that we're trying to target, point of care has become a very big, important one. That's the idea that you get a drop of blood, you can put it in a cartridge that goes into a device. You use a little bit of electrophoresis, and then you can identify if there is a presence of a pathogen, or there is a marker that tells you if you have an infection, or if maybe there's something else going on. In this case, the company that we're working with, they're already looking into some, they're doing some application work in the third world where there is sickle cell anemia. And so you have hemoglobin, and the sickle cell, the hemoglobin from sickle cell anemia will travel differently than the regular hemoglobin. So because of that, they can just look at two lines as you see there, and they can tell if someone actually has sickle cell anemia. Very simple way. And so we are working with this company just to enter the point of care market at that point. So really at this time, we have our product, we have developed it, we have a variety of different projects that we're focusing on, but we need to get noticed. And what we realized very early on is that it's difficult to get noticed by some scientists because they're working with what their parent researcher was working on and so on. And so we kind of have to just break that barrier. So we have focused a lot on social media now, webinars, just trying to inform people how our product works and why it's something that it matters to them. But with that in mind, we're also doing some long-term investment, which would be a student program. So when I worked with snails and I did a lot of protein assays, I had a really hard time finding antibodies. And whatever antibody I did purchase just took all of my scholarship money away. So Sandia BioTech has created an assistantship program for students where we will provide them with our reagents for free. If it's something that fits and it's something that they can use effectively in their projects, because ultimately in the long term, they are going to be the visionaries and they are going to be, well, later the pragmatists that will not change, right? And they will keep using our product. Any questions?
Any questions for Dr. Pena? Yes, sir.
Yes, so we do have we do have recently a patent for the fluoro bodies when it comes to the production of them. And we're working on a patent right now for the split GFP. Can give much more information on that right now.
Okay, thank you.
Yes.
Any other questions? I have just one question. Are you planning to sell these directly, or are you going to work through a distributor? How?
So we do have, at least for our split GFP, we do have a distributor. It's Sigma. And we've had some good traction with them, but we are looking to be noticed more out there, because what we realized from being part of the catalog of such a big distributor is that we kind of get lost in the midst of all the other things that are out there. And another problem is that the marketing is not as effective as it if it's not coming from you. So we're working on making ourselves more noticeable that way.
Thank you. Any other questions? If not, I want to thank the presenters for their great presentation. And I want to thank you for your questions. We appreciate that. So with that, I'll turn it back over to Greg, I guess.